biotechnology principles and processes, recombinant DNA technology. Now, there are some important tools which are utilized. How do we practice this recombinant DNA technology? The first important tool in this recombinant DNA technology are the restriction enzymes. Restriction enzymes. These are the important molecules, or what we call as the molecular tools for recombinant DNA technology. The second includes the cloning vectors. Cloning vehicles or vectors. Cloning vehicles, what we call as the vectors. These are the second important tools. One more thing is there. These cloning vehicles are of various types, which are used for the recombinant DNA technology. So far these tools are concerned. Restriction enzymes. Stewart Lin and Werner Arber were the first scientists who isolated these restriction enzymes in 1963. Stewart Lin and Werner Arber, which restricted the growth of these substances restricted the growth of bacteriophage in Bacterium E. coli. These enzymes restricted the growth of bacteriophage in Bacterium Escherichia coli. One of these enzymes added methyl groups DNA and second one cut DNA. The second enzyme was named as restriction endonuclease. That enzyme which could cut the DNA, that enzyme was called as restriction enzyme. Later on, H.O. Smith, K.W. Wilcox, and T.J. Kelly in 1968. After 5 years of the discovery, they isolated restriction endonuclease whose working depended on a particular nucleotide sequence. They isolated this enzyme from a bacterium. That bacterium is called as Haemophilus influenza. Haemophilus influenza. They isolated this from this bacteria, which is a pathogen. This hence it is called as HIND, H-I-N-D, HIND. Second, it was observed that this enzyme always cuts DNA molecules at specific place by identifying a particular sequence of 6 base pairs called recognition sequence. This enzyme has the capacity to cut because it can identify the recognition sites where the cut in the DNA fragment is to be made. This is the DNA fragment. It recognizes the sites where the cut is to be made. Restriction enzymes belong to a larger class of enzymes. These enzymes are called as nucleases. They are of two types, exonucleases and endonucleases. Exonucleases and endonucleases. So far, exonucleases are concerned, they remove nucleotides from the ends of the DNA. Exonucleases, they can remove nucleotides from the ends of the DNA. So far, the endonucleases are concerned, they make cuts at specific points within the DNA. There is difference. Exonuclease removes nucleotides. For example, this nucleotide chain DNA, they can remove here, they can cut here or remove here. This is the function of exonucleases at the end sites. But so far the endonucleases are concerned, they can have the specific cuts in the middle also. These are called as endonucleases. Thus, a restriction enzyme or restriction endonuclease recognizes a specific base pair sequence in DNA called a restriction site and cleaves the DNA within the sequence. Restriction enzymes are widely found in prokaryotes. They are widely found in prokaryotes. Prokaryotes are the bacterial cells. They are employed in prokaryotes and provide protection to host cell by destroying foreign DNA that makes entry into it. Here, they act as part of defense mechanism called restriction modification system. They form a separate system. That system is called a restriction modification system that acts as a defense mechanism in prokaryotes. In the second component is a modification enzyme. It adds a methyl group to one or two bases within the sequence identified by restriction enzyme. This is all about the tools of recombinant DNA. The first important tool are the restriction enzymes.